Okay, so this is for just specifically the short 5-week summer general biology Bio 121 class, and it's just a very quick overview. I already have information online. You have all the lectures and overview of the course and a variety of things. This is just to give you something really specific about the summer class. So it's short. It's only 5 weeks long, right? So normally this is 15 weeks of material. It is a lot of material to cover, even in 15 weeks. General Biology 1, if you don't know what it is, sounds like, oh, just an intro biology class, but it really isn't. A lot of the bigger universities use it as, I would not like to call it, because we don't call it a weed out class, but they do for people who are, say, pre-med, to tell them whether you're going to be pre-med or really not going to follow this track. At the other colleges, it's not viewed as that. It's just the first of two of a year of biology sequences, Biology 1 and Biology 2. Different schools divided up in different sorts of ways. The way most of us divide it is that Biology 1 is mostly cellular biology, and then Biology 2 is organismal biology. That's sort of how it goes. So for cellular biology, which is what this class then is as the first biology course, in a way, there's some logic to it. Cells are the smallest unit of life. All living things are a cell or made up of cells. Okay, so we start off studying life, biology, with cells. Cells are made up of molecules, and so you have to study the chemistry first. And if you don't understand chemistry or the chemistry of the cell, then you can't really study cells or anything that they do. So the way this course is broken down is that exam 1 is really sort of a very intro biochemistry. So it goes to some really basic chemistry. What are elements? Atoms. How are the electrons distributed? It's not chemistry heavy in the sense of the things that a chemistry class would emphasize. It's only emphasizing these sorts of topics that are going to be useful in studying other parts of the cell, the way chemical reactions happen in the cell, the way cells are structured. Okay, so there's some things that are very important in chemistry, but they aren't necessarily to us or in this particular course. Maybe somewhere else down the line they are, but we don't need them, so we're only going to cover the essentials, especially because we have a lot of topics to cover. Some courses cover a lot of topics and they cover them paper thin, very, very no depth at all. Other courses have very few topics and then they go deep into them. Unfortunately, this class wants you to do both. We're going to cover a lot of topics, and the expectation is that there's a lot of detail to them as well, which seems somewhat unrealistic, but that is just the way it is. So, and you only have in this summer class about 9 days for each block of information. So I say, study ahead. There's a little bit of self-pacing to this, but there are going to be specific dates for exams that you have to be ready for. And if you miss it, you're going to miss that assessment. All right. So what to do? All right. I'll give you the overview. 1 is biochemistry, atoms, molecules, electrons, how the different elements come together to start to form these molecules and how they behave. Mostly they're forming polar molecules to interact with water. So we're going to study water and polarity of water. Why is it polar? All right, how is it polar? What does that even mean? How does it interact with other molecules and how do they interact with each other? That then sets up the structure of the cell itself. So exam 2 then gets into the cell membrane. We're studying primarily eukaryotic cell biology, and so we're looking at the outer membrane of the cell, its structure, as a phospholipid bilayer with all these different chemicals that make it up, the chemicals that we introduce in exam 1. Then we look at the action and activities of the cell membrane, not just being a barrier between inside outside the cell, but inside the cell there's all sorts of compartments in a eukaryotic cell. So there's cell signaling pathways, there's communication, there's specific transport, there's storage. Exam 3 gets into energy, but it directly connects to the exam 2 topics because how cells store energy in a variety of different ways. One of the ways they store energy is in storing molecules in one space. A lot of them in a particular space, creating a gradient there. And then those things want to get out of that space, and when they do move out of that space, they release energy. So that the exam 2 topics then tie directly into the exam 3 topics, which is then going to be metabolism. So it's going to be enzymes, how do enzymes work, chemical reactions, and then the basics of glycolysis, pyruvate oxidation, the citric acid cycle, the electron transport chain, which is really a membrane, and proteins embedded in the membrane that push things from one side to another that then diffuse back. So then these things very tightly tied together. Exam 4, the last material, is genetics. All right, and so it's really broken into two parts of the genetics. There is chromosomal genetics. So what's a chromosome? What are genes? And then we go a little bit over mitosis and meiosis. But really, before doing those things, you really have to go into sort of molecular processes. Before those things can occur, DNA itself has to double up. Right? And so we have to go through the process of the structure of DNA, and then the process of DNA replication, the enzymes involved in that process and how they work. And then we get involved in gene expression. So essentially making RNA and then making proteins from the genetic information. And then how is some of that passed on to offspring? We go a little bit into the chromosomal genetics aspect and inheritance. Not a lot, but a little bit. Okay. So 4 lectures. I have recorded videos like this that cover all the topics. You have a list of all the topics in the class, and then you have links to all these videos. You will watch all the videos in a row. You can watch them multiple times. You can fast forward through parts that you know. You could rewatch parts that seem too fast. You can slow it down. You can watch them over and over, take notes on them, whatever you need to do to get through the material. But you have to do it. If you don't, you're just not going to, there's no point in taking the class. Really, you have to kind of go through that material. Now, I prepared slides for all these topics. I prepared them. I typed in everything that I would normally write onto a board. And there are pictures that are not just my drawings, but they're pictures from different textbooks, your textbook that you purchased, and a whole variety of other places that I have picked up over the years. So there's a lot of good resources there. I recommend printing that off and having it with you as you watch the lectures. In addition, you should have with you printed off the study guide, which you're using as a checklist. So essentially it's a list of terms and a list of topics. And you then should be able to draw and atom and put electrons in the shells, show a polar covalent bond between these two elements, and so on. And so as you go through the lectures, you have that checklist and say, oh yeah, I saw that. I haven't seen this yet. Where is it? So that'll be your clue to find out where is that information that I need, and I know I need it because it's listed here in that study guide / sort of checklist that you'll see. I give book readings. There are some links. There are topics in your book that you should just look up and use the study guide to look up and then read those specific things. I'm not going to give specific page numbers because it changes with the edition to edition of the book, and it's very hard sometimes because if you give you a page number, the topic doesn't start until really the middle of the page. And so I'm giving you topics. Look up the topic in the book, find it, read about that topic. It may only be a few sentences. It may be 5-10 pages. So that's what you should be reading, is the specific information relevant to the topics that we're covering. And how do you know that? You look at the study guide and you look at the list of terms. I also give links, which are a little more specific, to free online book resources. So you can go and just will bring you to a specific page or a specific topic. So you can look at that and just read that little bit. Usually they're very directed. They're not as in-depth, most of them, but they will give you some good information. How do you know that you're getting the information that you need before an exam takes place? Well, there are going to be quizzes. There's 16 total quizzes. There's going to be 4 roughly per exam. Some exams might have 3, some might have 5. So don't worry if you don't find all 4. But roughly, so I do 4 quizzes to prep you. They're multiple choice questions, which would be similar to some of your exam questions. If you do the quizzes, they're also a type of study resource. So they're going to cue you in on, I don't know this particular topic. It's on the quiz. I need to go look it up in the book. I need to go find the slides on that topic. I need to maybe find the lecture on that topic and watch it. And then I can go on and answer the quiz. You can repeat your quizzes. So you can do them 3 times. So do a quiz, maybe you get 6 out of 10 on the quiz. Find the information, redo the quiz. You'll get the average of your attempts. So then you get, say, 10 out of 10 on the next time. Now there's an 8 in the grade book. So 6 and 10. So that's kind of how that's going to work. The exams are going to be partly online and then partly sort of research projects that you'll have to, not a project, but topics you're going to have to research, draw, sketch, questions and problems that you're going to have to answer. Some things that you're not going to be able to just look up, things you're going to have to kind of know and problem solve to be able to do, right, and turn it in. There'll be a 2-day window, a timeline. So for each of the exams, it's given to you a couple different places online. So for exam 1, we're going to have June 1st and 2nd. So what that means is that midnight of May 31st, as soon as it just turns into June 1, the first exam will be available to you to do. Not that you're going to do it right then, but it's there. 48 hours it'll be available. And then after that time, at the midnight hits on the 2nd and it becomes the 3rd, it's gone. You can't do it anymore. And it's over. So make sure within that 48 hours you log in and do the online portion of the exam, and then you print off the problems and questions and do them. You're going to do those by hand, scan them, and then send them back in to me through the website. And it's going to be the same then for all the exams. So once you run through it for the first exam, you'll know what you're doing for all the rest. So this one is going to be then on also June, but it's going to be the 9th and the 10th. This one is on June 17th to the 18th. That's exam 3. And then exam 4 is going to be, we have a couple extra days there because it's going to be the final, the 26th to the 27th of June. That's also going to be cumulative, so it's going to have a couple questions from each of the previous exams, both on the online part and the written problems part. You'll have a few extra questions. These are worth 100 each, and this will be 150. So 100 on the genetics part, and then 50 more with a few topics of the others. You're also going to have a lab component to the course. There's a kit, which some of you have already picked up at the college. And you're going to be doing labs at home. I have a few of the videos loaded online right now, but just wait for the actual assignment to appear. There's going to be an assignment where you will submit the work through Blackboard back to me so that I can grade it. Okay, so wait until those assignments are active. There's about 9 labs that you're going to do, or some type of assignment. Some of them are going to be data I'm just going to give you, and then you're going to graph it. Other things are data that you're going to generate on your own and then have to do calculations. You're going to do some things at home. So the kit has some very simple things in it. It has dialysis tubing and ways to measure water. It has a digital balance in it that you can then weigh things for a variety of labs, making up molar solutions, and for the osmosis lab, and a number of different sorts of things in it. So you're going to do those at your own pace. They're not going to have a specific date for each one to be turned in. I'm going to post in the class, in the lab area, a guide to the suggested order that you do them in, and a timeline. So I try to have these 2 or 3 done by this date, and then try to have these done by this date. If you don't, those dates are not going to be due dates where you're going to be cut off or anything. You can do them all up until the end. But once the class is over, after the 27th of June, everything is off. You can't do anything, none of the quizzes, none of the exams. You can't submit any of the lab work. So if you wait till the end, there's going to be just way too much to try to do in the last week. Again, with only 5 weeks, you have to be really active every single week, probably every single day, doing something in the class, or you probably won't make it through all the material. Okay? So I'm talking fast because I wanted to keep this very short. And it's somewhat of redundant with other information that I have out there, but I just wanted to have something specific for the summer session, so that you know exactly what's going on and you hear it from me, and you know it's not an older video from a couple months ago. I'm making it right now. So email me when you have questions, and I will answer you as soon as I possibly can.